Okay, now it's setting up. Okay, now it's setting up on the YouTube site. Let me start with a brief introduction about today's speaker. So today we are very happy to invite Dr. Puska Kunwa and to give a talk on the 3D printing applied to biomedical applications. So Dr. Puska Kunwa is currently a postdoc at Syracuse University. So there he has been working on additive manufacturing with application to biological field. So he holds the master degrees in science and bioengineering from Temporary University of Technology, and he obtained his PhD in physics from the same institutions. Dr. Kunwa is actively involved in advancing laser printing technology, focus on development of multimodal additive subtractive printers with a remarkable microscale resolution for creating intricate multi-material constructs. Additionally, Dr. Kunwa explores existing and new techniques for 3D printing tissue constructs, incorporating biological interface and organized microvasculature. So he's also involved in the development of smart materials, particularly the creation of flexible and stretchable photonics and optoelectronics device using hydrogels, so offering versatile solution for various biomedical applications. His multidisciplinary approach and innovative research aim to address significant challenges in the field of biomedical. So then Dr. Kunwa, now you can start your presentation.
Thank you, Professor Genly, for the introduction. I would like to thank you for initiating and giving this opportunity to share my research. I'm also grateful to the organizer of this CFSM seminar series that will allow me to share my research work done both at Tampere University of Technology as well as here at Syracuse University. Please find my topic of the talk, different modalities in hybrid laser printing, HLP. Now we're exploring different modalities within this hybrid laser printer for advancing additive manufacturing. We're mostly focused towards the biological application and also development of new kind of 3D printers. So I'm affiliated, with the BioInspire Institute at Syracuse University, and the inspiration, of course, comes from the nature. Over the course of billions of years, nature has created, refined various structures, giving them unique shape, size, and function. Here's one example: blue color morpho butterfly. The blue color is not not coming from a pigment. If you zoom in, there are very, very small diffractive patterns at nanoscale dimensions. Now another example. So another example is your blood vessels and also lymphatic network, which is very, very complex multi-scale vessel network with a diameter ranging from few micrometer to few millimeters. Now again, if you go more further and go, if you see the growing section of the bone, then you see there, you see a arrangement of cell in certain gradient fashion. Now, recapitulating or recreating this kind of multi-scale complex structure is near to impossible using current technology. So now, with talking about technology, where do we stand? So compared to nature, our existing 3D printing technologies or techniques are very simple. They can print fairly simple 3D structures. These techniques can be divided into, let's say, additive approach as well as a subtractive approach. Now, additive approach, mostly, the first, this part is optical projection lithography. You can tell it a SLA, DLP clip, or projection SLA, DLP clip, or there are other like multiphoton polymerization, which is basically direct laser writing. Extrusion-based printing, inject-based printing, again, another form of direct laser writing of metal, selective laser sintering, or, for printing high-resolution electron beam lithography, etc. Now, the other approach is subtractive approach. This includes CNC machining, electric discharge machining, just remove, mostly used in metallic subtraction process, laser coating, water jet coating, as well as direct laser writing, another form of direct laser writing, which involves multiple photon ablations. Now, a few of the widely used techniques. So first is the, extrusion-based printing. So extrusion-based printing is a 3D printing technique. So where, the material is pushed through a nozzle, and, this create a continuous stream of a material building upon layer by layer to form a three-dimensional structure. Resolution governed mostly by this tip, and the resolution, let's say, around 100 micrometer and above. The other widely used is inkjet 3D printer. Similarly, works like your inkjet, printers by spraying tiny droplets of material in a layer by layer fashion until a complete 3D structure is formed. So the other is direct laser writing technique. So you have a focused laser beam, wherever, the laser beam shines the photosensitive materials, it creates pattern. The pattern can be in the form of polymerization or in the form of ablation. So basically, you can add material there or remove material there. Resolution goes all the way to 100 nanometer. Widely used, another is optical projection lithography. So basically here, rather than scanning, like in laser writing, you create a pattern of light, and that pattern is projected onto your photosensitive material, and you can print a single layer. Now iteration of this single, these layers can result in a 3D structure. So we are mostly interested in these two part of optical projection lithography as well as direct laser writing. Now, our photosensitive material is, just consists of mostly liquid photopolymer, which has combination of monomer, oligomer, and photoinitiator. So upon light exposure of suitable wavelength, this light cross-links, this light is absorbed by the photoinitiator with initiator polymerization reaction. So upon completion of the polymerization, the liquid resin solidifies wherever the light is exposed. So basically, you are turning liquid resin into the solid structure. Now, I would show here two work published by two reputed groups. One is the first one. This is done with the help of optical projection lithography. The other one is done with the help of direct laser writing. So both are the alveolus structure, which is used in air exchange, or it is a functional unit of your the smallest functional unit of your respiratory system. Now, if you see that scale here is 1 millimeter, this is fairly large structure as compared to the actual size of your alveolus. So this is almost like 10 millimeter, and each of these channels, which is, let's say, your blood vessel in alveoli is around 200 to 300 micrometer. Now, scaling down is almost like very, very difficult or near to impossible with this technique to get the resolution of, let's say, what the size of a blood vessel is, around like 10 to 20 micrometer. Now, there's another approach called, which is a direct laser writing. So basically here you have a collagen part, collagen material, and here you use a direct laser ablation to create a this alveolus structure. Now the size is very, very small, 100 micrometer. So basically this definitely gives the size of one air sac within the alveolus. Now with this, this is a serial scanning based approach. So to print this small structure, it takes already hours. Now scaling this up is now extremely time consuming. Now if you see your actual alveolus structure, which looks like this, the air sac, now this one small sac is almost around the size of this alveoli. Now if you want to print a whole alveolus sac, the whole alveoli, let's say, it would take almost a day or something just to print this less than 600 700 micrometer size structure with complete ablation. So what we did was we built hybrid laser printer. Now, to solve the scalability and resolution problem in the two different optics-based lithography systems, we combined these two techniques into one. So basically now we can leverage the scalability advantage of optical projection lithography that's able to print a relatively larger structure, as well as we can now use the high resolution advantage of direct laser writing. So these are the two modalities now used in this hybrid laser printer. So now this blue, I will not go in detail about this optical setup, but this part shown by the blue arrow is an optical projection lithography system, and the other one is the red dotted arrow is direct laser writing, can be used for ablation, also can be used for polymerization. So how does this work? You start with a liquid photoresin. Then this is a DMD, which is basically a digital micrometer device that's used to spatially pattern your laser beam. Then you pattern your laser beam and cross-link a structure. Now next you bring your femtosecond laser ablation or your direct laser writing beam and ablate structure within the the printed previously cross-linked structure. Now you do an iteration, you can complete the printing process. Now you could also do an additive approach with the same thing if you decrease the laser beam intensity. So basically now we have a hybrid laser printer, which is combination of projection stereolithography or optical projection lithography, then also a direct laser writing based on MPA, multiphoton ablation, and multiphoton polymerization. Now today I will talk about extension of modality 1, that is optical projection lithography. I'll discuss two projects here, MAPS and double network hydrogels, how we print based on this optical projection lithography, or how we develop a new technique called MAPS in this modality. In the second modality, I will talk about my PhD work where we use direct laser writing to pattern nanoclusters and how this nanoclusters is being used in serialization process. Now in third module, I will talk about, or I will combine this modality 1 optical projection lithography and direct laser writing to print some biological topologies. Let's start with the extension 1. So basically, optical projection lithography, and there we have built meniscus-enabled projection stereolithography. Let's dive deep into what exactly is this meniscus-enabled projection lithography. Just before I go into detail, just overview of what exactly is the optical projection lithography. Now you have a light that is patterned by your physical mask. Now this is brought into your wafer, which is mostly spin coated with a photoresin. Then you pattern that wafer. That's widely used in semiconductor industries. Now in 3D printing, we mostly replace this physical masks with a digital mask. Now digital masks are mostly created with they are used in this spectral light modulator or DMD, digital micrometer device. The mask in SLM, a spectral light modulator, or DMD, can be controlled using a computer-generated hologram, and masks can be changed at high frequency. So basically, in both of them, there are functional element which especially pattern the light pattern. For example, in DMD, there is like micrometers, which is size of like, let's say, a 10 micrometer. Then each mirror can be controlled with this computer-generated hologram. One form of optical projection lithography, which is a clip, which is widely used technique now as a projection optical projection lithography. Your company Carbon is completely based on this technology. So how this works, you have a vat, and there's a liquid resin fill in a vat. So, and there's an oxygen permeable window here. So oxygen permeable window, what it does is it makes a dead zone just below the fabrication window, so that the build part, the printing part, does not stick to the base plate here. And also, it creates a dead zone, so there is always a small gap, because oxygen kills radical responsible for polymerization. So now this at the beginning, the build, the support plate is all the way to the bottom. Then you start to project a mask, then shines a single layer. Then this as the this build support plate moves up, the mask continuously changes pattern. A new series of or new pattern of light comes, and at the end you kind of like get a complete 3D printing structure. You can print this structure fairly at the resolution of 10 micron. Now, projection stereolithography is type of bad polymerization. Now, advantage of this 3D printing PSLA is, the projection lithography is a better resolution, very fast printing time, stronger finished products, etc. There's also cons. There's like you have to use this bioresin, the resin for photopolymers, or photoresin, which are toxic environmental hazards. So you want to cross-link most of the resin, or almost all the resin. You don't want to leave any resin on the pad. So mostly utilize all of them. Next is, there's very little option for multi-material and gradient or composite printing because of the use of the VAT. So we come up with the idea of, we have invented and developed maps that addresses the, this disadvantage of PSLA. So let's see how this works. So this is a VAT-based projection lithography. Now, in place of that, we replace that with our planar PDMS slab. Then we added a pump inlet. Now pump. Now, rather than this acting as a reservoir, so basically MAPS relies on generation and continuous maintenance of the this resin meniscus form around a already printed structure. So as the print progresses, the material used up is replenished from the inlet pump. So this is a simple setup for the maps. So basically you have a CW laser beam that bring your pattern light all the way to the DMD, which patterns the beam and brings all the way to the fabrication area where you cross-link your structure. Then you have a pump for continuously pumping the material there. Now it is not a is a clip. It is not a continuous printing. It is rather discrete printing structure. Now, for the single material, you could do a continuous printing, but for the gradient, it requires to be, it requires to be a discrete step. So how, let's say you have a 50 micrometer, you want to print a 50 micrometer layer. So what happens is this moves up, drawing enough material for printing 50 microns, then it goes down 150 micron layer, leaving a gap of 50 micron, then it cross-links. Now the laser will turn on and cross-links the 50 micron gap. So this iteration will complete the whole structure. Now, just to avoid the confusion, in this work, we have used different kind of arrangement or configuration for inlet tubes. So it can come from side, it can come from top, or it can come from the bottom. You will see different configuration in different experiments as this representation progresses. So let's start with single material printing using maps, using this, let's see the video for the single material printing using the maps-based technique. So it is not a continuous, it is a discrete process. So here's inlet tube, and here's somewhere here is the pump, which is continuously flowing the resin to the fabrication area. I'll purposely stop it here just to show you. Now there's a breakage of flow. Okay, this see this carefully, because this concept comes later on. Now there is a breakage of flow, so I had to increase the pump rate. So now again the material flows into the fabrication area, and the printing continues. So this is Statue of Liberty printed, being printed. And here are a few of the structure printed with the maps technology. The first one is a lattice structure, Statue of Liberty, human heart-shaped structure using, these are all printed with PEGDAS 250 resin. The resolution of printing is around 10 micrometer. Now we can do a multi-material printing using maps. See multi-material printing. Okay, just a little bit of background. There's two tubes, one tubes for one material, that another tube right at here as second material. So once we kind of flow a desired amount of resin that is printed, and almost all the resin is consumed, then we'll flow the another resin. So this helps us to print a lateral multi-material in vertical direction without involving a cleaning steps. Now, once the gray resin is completed, now another resin will flow there, which is again transparent PEGDAS 250 resin. Now the size of this structure is, the diameter right at here is around like 5 to 6 millimeter. You can see most of the resin is consumed in this process. So this is the printed structure, again at the resolution of around 10 micrometer. Now, the cleaning steps of maps is fairly very simple. To see a video how this is done. Now, for lateral multimaterial printing, you need a cleaning steps. So when you print the structure, almost all the resin is used up. Then you can just wipe that resin, then bring this back, then throw another material and start printing the structure. With these cleaning steps involved, I mean, right now we have not automated it, done manually, and the cleaning is done manually. In the future, we do plan to automate this process. Here are some intricate structure which shows, both lateral and, vertical multi-material printing. Now in maps, one of the main advantages is gradient 3D printing. Now just the concept of that meniscus again. Now you have a PDMS, let's say this gray region is your PDMS slab. Now if you drop a resin based on your contact angle, it is going to make a dome-shaped structure. Now this is defined by a contact angle θ1. In our case, with our resin, it was mostly, the θ1 was mostly like 75 to 95 degree. Now, as the print progresses, what happens is another contact angle is formed between the printed parts and the liquid resin. Now that's defined by the contact angle θ2. So here we found our contact angle was around 20 to 30 degrees. So now this because of this two contact angle, now you have this meniscus, which has certain volume of your resin, which is given at this figure. So basically these volumes are in, and this is used at the reservoir in maps rather than the whole vat, so that it is very, very easy to change the resin as the print progresses. Now here we have a two fluidic pump and an inline mixer, again printed with the help of maps technology itself. Now inline mixer, you can have different fluid rate and you have different kind of desired composition coming out from this inline mixer, and it is guided all the way to the fabrication area. So this is how the maps used. Now, ideally, for printing a gradient structure where the resin composition changes each layer, you want the resin volume to be ideally equal to the volume required to print a single layer. However, this is not the case. The volume is much more higher, again defined by the contact angle, this θ1 and θ2. Now you could try to control this volume by lowering the pump rate or flow rate. Now, however, you saw that in the video one, this could discontinue flow as you decrease the pump rate. So if it is discontinued flow, then there will no resin will not reach to the fabrication area or to this meniscus region, and this will be the structure will not be fully printed. So that way you cannot decrease the flow rate from here. Now the other one is what you could do is you could change the play with the contact angle, try to decrease the contact angle and increase the high wettability within the photopolymer within the PDMS layer. Now we worked with Professor Jen Lee and this simulation where we saw that for a higher contact angle, it is more obvious that you will break this flow and result in a discontinued flow. But if we decrease the contact angle, then it's much more that the resin will spread, and it can easily reach to the fabrication area. And also now that way, if the contact angle is lower, your volume in the resin is decreased. Now how do you decrease or increase the wettability or decrease the contact angle? So what we did was we did a plasma etching. So basically plasma etching process decreases the contact angle and increases the wettability. For example, we did this pattern, a cross pattern structure and plasma etch. So basically now this is just a slab. So basically there's a pattern which this is a hydrophilic area, this is a hydrophobic area. Now water will contain only in this hydrophobic area, so that way you can distinguish higher and lower wettability area. Now with this approach, we created our we patterned or etched the wettability area, higher wettability area, and did a gradient PSLA, and let's see how this works. Now we are using the top configuration of our inlet flow. Now here's somewhere here is the mixture. Now the liquid is flowing from here and guided towards the fabrication area using that each region using plasma etching. Now the meniscus volume is very, very low. Now you can see the gradient structure coming out from the build, or you stick onto the build plate. Now you can see a complete gradient structure where the material properties varies when it comes from top to bottom. Here's the one example where there is a variation in color gradient printed with maps. Another more example is now this is the example where the gradient properties changed in the gradient is in mechanical properties. For example, there you have a more steep structure. As we go up, you have a less steeper or softer structure. So more intricate structures, front view, backward view. Now gradient in exponential. We can also do a cell gradient, basically within your inline mixture, where you can add one cell solution, the other one your bio ink, and the mixture mixes and bring it to the fabrication area. Then there you can print, and this is a confocal image. So basically green dots are live cell and red dots are death cell. So you can see some sort of gradient formed within this printed construct. Now you could also do some add some magnetic material in a gradient fashion and see the response. For example, in this one we use 10% Fe3 throughout the construct. The second one we varied the gradient, gradiently the magnetic nanoparticles. In third we did not use anything. So basically we saw the magnetic response and saw that in the form of angle. So you can definitely see different magnetic response based on the distribution of magnetic particles. So you can create even more intricate structure with this approach. So this paper is under review in Advanced Material, and you can already see that access this paper in bioRxiv journal. So this is one. Now again, extension of the modality one is still the PSLA. We worked on the double network hydrogels. So let's see the next topic on this extension of modality or extension of PSLA. So we use our PSLA to print double network hydrogels. Now, hydrogels are widely used in biomedicine, so basically it has found applications in drug deliveries, tissue engineering, biosensing, flexible electronics, soft photonics devices. But traditional single network hydrogel, they exhibit inferior mechanical properties, which limits their use in the field. Now there's a research interest growing on these double network hydrogels, which have superior properties such as toughness, stretchability, compression strength as compared to single network hydrogels. So inspired by this work from Jung-Yoon Sean, published at Nature in 2012, so we aim to fabricate 3D structures using PSLA for elastomeric optical components based on these hydrogels. So here they use acrylamide one, which is photocross-linkable, and the other network is alginate network, which is ionic cross-linkable. So we aim to make optical components based on this printing these hydrogels again. So now for your PSLA, what's required is your liquid has to be in solution form. But as soon as you add, for example, sodium alginate is an ionic cross-linkable. As soon as you add a calcium ion within the sodium alginate, then it readily cross-link forms a gel. So there you cannot use that. Now you can cast it, but you cannot really 3D print it, especially with projection stereolithography. Now what we come up with, the idea was, we added acrylamide, MBA, crosslinker, and photo initiator lab. For these are required for photo crosslinker for acrylamide. Now sodium alginate. Sodium alginate was just left there. We did not add calcium chloride so that the sodium alginate does not, the ionic cross-linking doesn't occur. Now then we fabricated photo crosslink the structure, acrylamide structure. Now this is only a single cross-linking, photo cross-linking at this point, which is still weak. Now later, sodium alginate is still in the sample, or is still in the cross-linking, but on cross-linkable. Now it's not cross-linked, still in the somewhere embedded within this acrylamide, but not cross-linked. The network is not cross-linked. Then later we dip this whole structure into a calcium chloride solution. Now the calcium starts to fuse inside. Now when you go inside, then it starts to form a another network. So this way we can enhance the mechanical properties of this printed structure. Here are a few of the printed structure with a resolution of 200 micrometer. But one of the thing what we found was when we dip this into the calcium chloride solution, our structure turned into into the opaque. Now our aim is to make a optical component that your structure has to be transparent. So now this was the first report of shaping double network hydrogel using PSLA, using this both ionic and photo cross-linking method. So we published this in ACS Applied Materials and moved forward for the next project where we want to create a transparent 3D structure now. So we replaced that with a copper carrying, and the alginate was the one which was giving us a that opacity or translucent properties. So we replaced that alginate with the copper carrying, and so what we found was this is a polysaccharide and mostly extracted from the algae, and this has a properties called thermo cross-linking. Now above 80 degrees Celsius, this is a solution. Now below 80 degrees Celsius, this forms a gel. Now we still use that acrylamide, LAP and MBA as a cross-linker, which is photo cross-linkable. We only change copper carrageenan. Now to make this, you know, this whole gel or whole photoresin into solution, it has to be heated to 80 degrees Celsius. So we made a, you know, sample holder with using the heat plate and copper, which maintains a temperature of holder to 80 degrees Celsius. So while printing, it is, you know, it remains solution so that we can print the structure. So this is all this stereo technique is called a temperature control positive stereolithography of tops. Based on that, we printed you know, the 3D structures in more intricate 3D structures using this one. Now in the tensile region, it does, you know, it stretches almost like 11 to 12 times. The strain is 11 to 12 and the stress is around 200 compared to its parent gel. You know, it has way better mechanical properties, but you know, we did see the necking behavior in this one, but we did solve the necking behavior by simply dipping the structure in immersing the structure in the water bath. Now here's the example how we solve, you know, how the you know, the tensile performance of dog bone created worked when we just dipped and on, we just remain in air before doing a tensile test. So basically, you can already see the right here necking properties. Now this remains, you know, there's no necking here. So the mostly stretching is happening at the necking portion. Now here is uniformly stretching. So basically now when you dip that into the water, what happens is, you know, this is the one, the stress strain curve. Now when you dip that into the water, what happens is it requires much less stress, so you lose the, you know, toughness of this structure. But what we can do is, while you prepare the sample, you can already first decrease the you know, amount of water, then later dip that into solution, so that when doing this red curve, which is this structure, so basically the water concentration is 80%. Now when we do the tensile testing for this structure, now after the immersion into water, it absorbs water and the water concentration, effective water concentration, goes above 90%. So basically now if you prepare the sample with water concentration only to 60%, then later dip that into the, you know, water again, then the effective will go to the 80%, which is similar to the one we did for this original ones. So basically you can restore the stress, you know, of this or mechanical properties of this or toughness of this structure. Now this not only works best on, you know, tension, but also on compression. So basically we printed up you know, Mayan pyramid and compress this to almost like a strain of 90%, and it is still you know, regains its shape. It's better than, you know, our compression stress strain test says that, you know, our hybrid gel is better than its parent gel, carrageenan and acrylamide. So need almost like 15 MPa to crush the structure to pieces. Optical performances, now it's fairly transparent, like more than 90% in the wavelength range from 400 to 800 nanometer. And we printed an hexagon lens and we created a bezel beam with the help of hexagon lens marked by this annular ring. Now we also made a manual stretcher to see the tunability of this lens. Oops. So basically now you are stretching the lens from four sides. Now when you stretch the lens, your annular ring of the basal beam starts to decrease. So we did see a proof of principle of optical components using this TOPS based printing. Now another extension is now modality 2, where I did a direct laser writing of silver nanoclusters. So basically now I'm using a direct laser writing the portion marked by the the red arrow dot. So contains both MPA and MPP. So direct laser writing. So basically you start with the resin, then you shine light wherever you shine light, you cross-link that. Now once you complete the cross-linking, then you remove the remaining resin, photo resin, and what's left is your structure. This approaches of scanning, it can be raster scanning, vector scanning. One of the work that I did in my master thesis, where I printed a micro logo of my university, Tampere University of Technology. So now you not only do a cross-linking, but you can also ablate structure from a precise location. Now we did a direct laser writing of nanoclusters. So what are these nanoclusters? So basically these are very, very ultra small nanoparticles with size less than 2 nanometer. You can call this as a, metallic quantum dots also. Now the size is less than 2 nanometers. So there's not enough electron for collective oscillations, such as and for the properties and for the plasmonic properties. So more like there is a discretization of energy labels. So basically you can see there a transition happening. So the one of the unique properties of these nanoclusters, like metallic in semiconductor quantum dots, they have a strong fluorescence emission. So it's synthesized in 2 steps. One is reduction. Now you mostly start with, let's say if it is a silver, then you start with the silver ion. Now silver ion, when you reduce that with light or any other reducing agent, what happens, it becomes Ag0, then they start to coalesce. Now if you don't control their growth, then it becomes nanoparticles. But if you control growth with the stabilization metrics, such as geolites or certain polymers like PMAA, then you can restrict this growth to a nanoclusters and get these properties. For example, this is a silver nanoclusters in PMAA film. So basically they start with a silver ion and PMAA, then shine light to reduce. After that, these nanoclusters are formed. Now what we did was we take this PMA, polymethyl acrylic acid, and silver nitrate, and spin coated thin films in a microscopic cover slip. Then we expose the the film to see if we can form a nanoclusters or not. So it's a video. Now at around like 1 minute or so, you will start to see the formation of nanocluster marked by strong fluorescence. Now you can see the nanoclusters forming. Now as the nanoclusters form, now you start to now see the formation of nanoparticles also starting from the center of this spot. Now you start to bleach to reach to almost minimum. All of these nanoclusters bleaches as the time progresses. Now this is the summary of that video. Basically I took the photographs at the different time points, at time points just at the beginning, then at the point where the fluorescence was maximum, then at the point where the after most of the nanocluster has bleached and there's nanoparticles formed. So here at the beginning there's just a silver nitrate, so just a silver ion, so no fluorescence, no Raman, or very, very minimum, basically stray lights. Now second, there is a high fluorescence and enhanced Raman remarked by this cyan curve here, which is a fluorescence intensity curve. Now as the time progresses, you start to form a nanoparticle, so basically decreased fluorescence and decrease in this Raman signal as well. Now we can form a nanoclusters. Now we have used these nanoclusters to, pattern or do a direct laser writing on a thin film substrate. Basically, we use 780 nanometer to reduce this focused laser writing, employing two-photon polymerization to write complex structures like I mean, this is a fairly two-dimensional structures, very small. I mean, this is a Tampere University logo, and this is Aalto University logo. This was our collaborators' work. We did, some emission and excitation spectra and broadband. We do see a strong fluorescence with the broadband emission. And we did some Raman signal, was mostly this Raman signal was coming from carboxylic acid from from PMMA polymers. Even some hot spot within the written lines. So basically, on written portion there were no Raman signal at all. Now, so basically now we use this direct laser writing to create a silver nanoclusters, and this work is being used by a startup called Fluorotus. So basically, let's see the video how they are using this this technique. Well, I'm not sure if you guys heard the sound or not. If not, then you can go to YouTube and just type Flow Trust, and as you can see the video there also. So now that was that, the end of the modality 2. Now the third is the modality 3, which I'll quickly complete this one. So I use this hybrid laser printing to print this intricate scalable and biological topologies. Here we mostly use PSLM mode as well as MPM mode. We have not used MPP, or we only use MPM. It's ablation mode, not the polymerization mode of direct laser writing. So here how it works, we have a CAD design. So that CAD design is sliced into a model, into a digital mask. Then you print, let's say, our aim was to print a pyramid with a embedded, let's say a cuboid structure. Then you print half of the pyramid using a PSLM mode. Then you bring a femtosecond ablative, iterate this additive and subtractive steps, which is an automated step. Then at the end, where completion ablation, you complete the structure. So basically, this is the result. Now, these cubes here you see is ablated like 2 centimeter deep inside the structure, so you don't have limitation of now laser penetration in ablation using this technique. So this was done in model PECDAS 700 hydrogels. And we have mostly used this high laser printing in printing microfluidic chips, again in PEGDA 700, which is not a bio-friendly, but not a biotoxic. It's a biological inert material. So we did sow some of the microfluidics with different types of complex channel or even like 3D channels. So basically, we also did some cell communication chips and cell migration chips where osteocyte, we saw osteocyte communicates with each other, or there's a SOS, cancer cell can migrate through this small channels, which is fairly like 7 micrometer. Now, based on this work, another startup microfluidic 3D has been established to commercialize this not only this product but also this technology, hybrid laser printer. Now this is an ongoing work, and now we are looking for a material system for HLP which are more like bio-friendly. Now we're looking for a material which is more like works on additive as well as subtractive, as well as they are biocompatible, meaning we are mostly working to create a multi-scale blood vessels. So let's say we're looking where the material where there's not just biocompatible, but also they adhere to the surface and also proliferate within the surface. So because this is unpublished work, so I have kind of like not shown what exactly are these materials, but we have found like a couple of materials that works for us. Basically, these are mostly formulations of 2-3 different kinds of bioinks. So we mostly stick with this, let's say a 16 model bioink. Now, here's the example where it works at additive manufacturing. Everything is created with additive manufacturing. The scale is fairly large, 5 millimeter. Now it does work with ablation. So this is ablated just inside, 250 micrometer deep inside the structure. Fairly like less than 10 micrometer lines or channels. Now this is the example of the third portion where the cells are adhering to the surface and they reach to the confluence within 2 days. So basically they are happy there, they are proliferating within the cells, so they are dividing there. Now we did so some of the channel circuits using this material system. Basically, we're trying to create a interface which is relevant to your alveoli structure, where there is a very close interface in blood and air barrier. Also, this is this kind of interface is also found in functional unit of kidney. The nephron. Inside the nephron, there is a Bowman's capsule. There you can find this kind of interfaces. So trying to create those kind of interfaces, or more intricate structures like alveoli structure with size scale. For example, the air sac portion is done with the help of PSLA, and the outer part is done with the help of laser ablation. And green dye, Fitzy dye is flown through this, and you can see a nice fluorescence image of this complex structure. Now we're mostly interested in creating a multi-scale blood vessel. First, we started with additive approaches. So basically, now we created this kind of the channel. Now within this channel, if we zoom in here, after you insert or 2H11 cells, which is the endothelial cells, which makes a nice lumen in this 3D channels. So here's the exam fluorescence image after the stent for actin and nuclei. So basically, now if this is the endothelial or a bottom view of the endothelial channel. Now if you see the 3D section right at here, then you could see a nice monolayer forming formation, because of the cells are making a nice monolayer, making a good lumens of the structure. Now we are also starting to make more complex structure. For example, this is created with the PSLA, then these are joined with the direct laser writing. So we do see some endothelial cells now going on, is starting to form a monolayer there too in the smallest channel. So I have only reached to this point here, so more to follow in the future. And thank you for your attention. And I would like to acknowledge my supervisors and all my co-authors, collaborations, and funding agencies. Thank you very much.
So thank you, Dr. Kunwa, for the very interesting talk. There's a lot of a lot of different applications.
Yeah.
So any question from our audience? So maybe I can start with a few for my question.
Okay.
At the end of your talk, you mentioned that you can do the 3D printing about the blood vessels. So there's a lot of endothelial cells is in the network.
Yeah.
So how you can make sure, because cells need some oxygen or other stuff to survive. How you can make this network can keep the
Okay.
cells still?
So basically, we can now, right now, we are not flowing that thing. So basically, we are flowing, they are dipping this into a media which has all of this, considering, food for cells, let's say, even like, within this gels, it is not a compact gel because there's like, what is called, let's say, compartments within this hydrogels. So
Yeah.
you already have an oxygen and other things. So there and also protein for attachment. So that's where how the cells are alive. I think I answered your question. I don't know.
Yeah. So usually how long you can keep the printed blood vessel still functional without
Okay, it is just a very initial experiment. So to be true, we have not. I would say like 6 to 7 days. Then it start to they start to fall apart. So
okay.
I mean, there's a lot of work has to be still done there. This is the starting point.
Yeah.
Now, if you see most of the study, they mostly report this kind of work for 5 days or 6 days. So what happens at the 15 days or what happens at the 30 end of the 30 days, they don't never report. So yeah, we have observed that, they start to disintegrate at some point.
Yeah. This is very interesting because you put the cell in the network and the cell actually they if you put some, for example, in apply some flow condition or you apply some nitrogen or other stuff, the cells can the network actually the vascular can evolve over time because of the
Yes,
cell are alive there, right?
right.
It
Yeah.
will be very interesting for the follow up or research on this
Yes,
topic.
absolutely. Yeah.
Another question is when you use your nanoparticle and do the printing, you said you can create the very small the QR code. I noticed
Yeah.
that the scale is 5 micrometer. So usually if you print this in any, for example, the passport or any other ID, actually we cannot see that, right?
Yeah, we cannot see with our naked eyes, right? So these are very, very small, let's say 10 micrometer. You can go all the way to 10 micrometers, so or even smaller than that. And it has a very unique, if you see the fluorescence signal, that is not a uniform signal. There is a Raman peak which is peaking at the fluorescence
Yeah.
curve, makes this very unique emission signatures. I think we don't find this kind of, at least I have not seen this kind of emission signature in any of the other material.
Yeah. So if you want to see the pattern, you have to use some a special device to see that, right?
Absolutely. Now again, if you you have to make a addition of let's say a mobile a cell phone based fluorescence microscope or cell phone based fluorescence spectroscope, that has been, I mean, the work at Tampere University, they are working on that too. So right, especially if it is very small, but if it is bigger, then you fairly see. But again, if you want to see the emission signature, you definitely need the emission spectroscopy.
Okay. Okay. So my last question is about the multi-material hydrogel printing. So you mentioned that you can generate the hydrogel, use the printing method, and you can do the tensile loading, compression, and loading. And
Yeah.
have you have you considered because now you have the 3D printing technique, you can have fully control about the structure of the hydrogel. Do you consider the fatigue performance of these hydrogel materials?
Oh, well, we have not worked. Really, what do you mean by fatigue?
This means you do the circuit loading, so how the defaults accumulate inside of the material or the
So,
aging effect.
okay, so basically, do you mean like on double network hydrogel or on the, maps printing or double network hydrogel?
The double net
Okay,
hydrogel.
yeah, we did, fatigue several times, and I did deleted a slice from here because it was, being long, but we did have a result on that front. From first cycle to second cycle, there is very small fatigue. Okay, but from third cycle to 100 cycle, it is just remains the same.
Okay, so is there anything you can do to improve this performance, or
No,
if you use?
actually, there's not much of fatigue, less than I would say 5% or something like that from first to second loading. So I think we don't really need to work on that. It's
Okay,
working very well. Yeah. I mean, if it is a, compression, or if it is a tensile, let's say if you are compressing to 80% or 70%, even you don't observe the fatigue. So if you go below like 90%, then only you start to observe that, fatigue resistance.
okay, yeah, very good performance of
Yep.
this kind of material. Yeah.
Thank you.
We also have about 5 people on the YouTube side, but they cannot ask a question. So
Okay,
for later,
gotcha.
maybe they have questions. They were contacting you by email.
Yeah, absolutely. They can always contact me by email. Yes. I'm always, happy to help anybody, happy to answer any questions or concern, even, suggestions or, even collaboration. If you have, some new ideas coming out of this work, then you can always contact. We could, initiate collaboration like we have done with you, so
Yeah,
has been
yeah.
a very fruitful, collaboration.
Okay. So any question from other audience? Ani? Okay, if not, we will end it here. Thank you very much,
Okay.
Dr. Kama, and we will keep in touch in the future.
Okay. Yep. Thank you very much for giving this opportunity. Have a good day.
You too. Bye.
Bye.